Vanakkam. Welcome to this course on classics in neuroscience. We have been looking at classic Nobel Prize winning works and we will continue our discussion of classic Nobel Prize winning works of Edgar Adrian. In this video, we will look at how Edgar Adrian's work essentially helped to map the cerebellar cortex, and he studied the common features and differences across species and the relationship between brain and behavior, and his contribution in the studies of olfaction and the olfactory system. So, Adrian showed that different cerebellar regions were associated with the forelimbs, hind limbs and the head. He recorded from groups of cells and single cerebellar units in cats, monkeys, dogs and even a goat. And he showed that different regions in the cerebellum were associated with different limbs, whether they were forelimbs or whether there was a hind limb or head. Now we know that there are these maps in cerebellum. Actually, there are multiple maps. We now know that there are multiple maps in the cerebellum. The details of this is beyond the scope of this course. These are called as somatotopy in cerebellum. Somato means body, topy means map, a body map that is found in cerebellum. There are more than one map of the body found in the cerebellum. Do check my other course in which I provided detailed description of these maps in the cerebellum. He also found multiple somatosensory areas in the cerebral cortex, including primary and secondary representations, and he found that the secondary representation was similar to an animal lying flat on its back with its head pointed anteriorly. This was his mapping of the cerebral cortex. Earlier one was about the cerebellum. And he found that there are multiple representations that he could have. There are different cortical representations, and this topic is still being debated. This is beyond the scope of this class to discuss this. I have discussed some 25 different scientists and their contributions towards mapping of the cortex and their representations in my other course on neuroscience of human movements. But here Edgar Adrian essentially theorized that each area likely has a different function. So, there is functional localization that Edgar Adrian posited, and this is still being investigated and examined. Edgar Adrian mapped surface of brain in several species, including cats, dogs, monkeys, pigs, goats, sheep and even a Shetland pony. He used natural stimuli like rubbing of the skin, stroking with a feather, pressing the skin with fingers and other such natural stimuli, and wanted to record the response from the brain. And he found that different hemispheres receive inputs and projections from the opposite side, the different sides of the body, and body surface appears upside down, facing forward in the primary sensory cortex. That is, the body is lying flat with the face or the head pointing anteriorly or to the front. The size of the cortical representation depends on the importance of the part of the body in various across species. Remember, this was also around the time when Wilder Penfield did similar mapping in 400 plus epilepsy patients, and he in humans, that is, and his maps were the motor maps. Well known homunculus are the Penfield maps that we know of. But here this is about not only restricted to motor maps, but somatosensory cortex, for example. And Adrian believed that he can predict the behavior or function of the animal physiology by observing their behavior in the wild. He could predict the physiology of the brain by observing the animal in its natural habitat, and studying physiology could help understand the behavior of the animal in its natural habitat. That is, if I can predict behavior, or if I can observe behavior and get insights into physiology, then by studying physiology I can also get insights into behavior in the natural habitat. Adrian also dabbled with studies of olfaction. He studied olfaction because olfaction was a neglected area of research, but in animal life it has a crucial importance because olfactory organs are very large in many animal species, not that large perhaps in humans, but the other animals, in particular dogs for example, have huge olfactory regions, and there is variability across species that he could find in response to different odorants. That is, sensitivity of the olfactory system varied across species. Rabbits for example are sensitive to grassy and fruity smells, whereas cats respond to foul smells and odors of other animals. So, essentially these animals and their response to different odorants essentially suggested what their behavior is in some sense, in some broad sense. He also observed specificity within the olfactory system, but as the stimulus strength increased, the coding or the olfactory system complexity increased in a very big way. So, patterns of activity that he measured across neuronal populations play a critical role. That is, it is not individual neurons, but rather several populations, several groups of neurons that together perhaps encoded this complex behavior, complex sensory input. With this, we come to the end of this video. In this video, we looked at how Edgar Adrian essentially helped to map the cerebellar cortex and the common features and differences across species in the mapping of cerebral cortex, in particular the somatosensory cortex, and the relationship between brain and behavior or neurophysiology and behavior, and Edgar Adrian's early studies on olfaction and the nature of olfaction in different species and how it varies across species. With this, we come to the end of this video. Thank you for your attention.